In this topic, we're going to discuss the translocation of organic molecules in plants. So we're going to look at what is the source and sink, what is the mechanism of translocation. So we're going to look at the transfer of sucrose into sieve tube elements from photosynthesizing tissue, mass flow of sucrose through sieve tube elements, the transfer of sucrose from the sieve tube elements into storage or other sink cells, and then we're going to look at an experiment using an osmometer. Evidence for the movement of solutes in phloem, and then finally evidence for and against the pressure flow theory. Now, having produced sugars during photosynthesis, the plant needs to transport them from the sites of production, which we call the source, which is generally in the leaves, to the other parts of the plant known as the sinks, where they'll be used directly or stored for future use. Now, sinks can be anywhere in the plant, sometimes above or below the source. This means that the transport of phloem occurs in any direction. Other organic materials to be transported include amino acids, and then the ions, potassium, chloride, magnesium, phosphate, nitrate. Right, let's have a look at the mechanism of translocation. So the rate of transport in phloem is too fast to be explained by diffusion. This means it must be an active process requiring energy. It also occurs in large quantities, and there's still some resistance to flow. Now, what is in doubt is the precise mechanism of how translocation is achieved. Current thinking favors the pressure flow hypothesis. So there are three phases. The first is the transfer of sucrose into the sieve tube elements from the photosynthesizing tissue. The second is mass flow of sucrose through the sieve tube elements. And then lastly, the transfer of sucrose from the sieve tube elements into the storage or other sink cells. So we're going to look at each of these in detail. The first one is the transfer of sucrose into sieve tube elements from photosynthesizing tissue. So sucrose is manufactured from the products of photosynthesis in cells with chloroplasts. The sucrose diffuses down a concentration gradient from the photosynthesizing cells into the companion cells. Protons, which are hydrogen ions, are actively pumped from the companion cells into the apoplast, which are the spaces within the cell walls, and this uses ATP. The protons then flow down a concentration gradient through carrier proteins into the sieve tube elements. Sucrose is transported with the protons, so this is called cotransportation, through the cotransport proteins. Sucrose can now diffuse through the plasmodesmata into the sieve tube elements, as you can see on the left there. So here you can see the cotransportation of sucrose and protons. Firstly, the protons are pumped out of the companion cells into the apoplast. As you can see, this creates a proton gradient where you have lots of protons in the apoplast. So the protons can now diffuse back down their concentration gradient. And as they do this, they bring sucrose with them. So this is called cotransportation, and this occurs through a special protein called a cotransporter protein, and in this case it's a sucrose proton cotransporter protein. So let's move on to the next phase in translocation, the mass flow of sucrose through sieve tube elements. Now, it's important to understand this part. The sucrose produced by the photosynthesizing cells causes them to have a lower water potential, as you can see at the top there. Water, therefore, moves into them by osmosis from the xylem. So have a look at that blue arrow, which has a much higher water potential. Then at the sink at the bottom there, the sucrose is either being used up during respiration or converted to starch for storage. So these cells now have low sucrose concentration, so they've got a high water potential. So at the source, there's a low water potential. At the sink, there's a high water potential. So at the sink, at the bottom there, water moves from the sieve tube element into the sink cells, and it also goes into the xylem vessels down its concentration gradient. So as a result of water entering the cells at the source, at the top, and leaving at the sink at the bottom, you've got a hydrostatic pressure being created. This creates a mass flow of the sucrose solution along the phloem, so that middle part there. Now this is a passive process, this mass flow. We're going to look at an experiment to demonstrate this process in a moment. Okay, the last phase is the transfer of sucrose from the sieve tube element into the storage or other sink cells. So the sucrose is actively transported by the companion cells out of the sieve tubes and into the sink cells. So it's an active process requiring ATP. Okay, this model here is called an osmometer. So if you look at this model to demonstrate the movement of sucrose solute by pressure flow in phloem, you've got cell A and cell B. Cell A has a membrane that is permeable only to water. It contains a solution with a high concentration of sucrose, so it represents the source of the plant. Remember that at the source, sucrose is loaded into the sieve elements by active transport. So the water potential will be low. So this whole diagram here is an osmometer, and it's made of two permeable containers joined by a tube. These containers are filled with different sucrose concentrations and placed in the water, so you need to identify which is the source and which is the sink. If we have a look at cell B, this has also got a membrane that's only permeable to water. However, it contains a solution that has got little sucrose. So it's the sink of the plant. So the sink is your roots or respiring tissues. What is the water potential? The water potential is going to be high because the sucrose is being converted into other compounds, for example, starch, if that was the case in the plant. So the water is going to move out of the cell. So on the left is the source, high concentration of sucrose. On the right is the sink, low concentration of sucrose. Okay, let's have a look at what's going on. If you look at cell A, water enters the cell by osmosis because it's got a lower water potential than the water surrounding it. This creates a turgor pressure as water moves into the cell by osmosis. The sucrose solution then moves by mass flow through the glass tube, which represents the phloem. It arrives at the sink, which is cell B. The water will then leave the cell because of the initial high pressure potential. And as the water leaves, this causes a low hydrostatic pressure. So this whole process will continue in a plant because you've got sucrose being continually produced at the source and being removed at the sink. Now in this experiment in the classroom, the process will eventually stop when the pressure throughout equalizes. Okay, let's have a look at evidence for the movement of solutes in phloem. So a few scientists have a few ways that show that sugars are carried in phloem and not other tissues. The first is using labeled carbon in carbon dioxide. So what you do is you have carbon dioxide that has C14. If you give a leaf this carbon dioxide with light, as you can see in this diagram here, this carbon dioxide will be converted into carbohydrate so that C14 will be incorporated into the carbohydrate. Then, if after a few hours you take a section of the stem and you cut it, and you use photographic film and place it over the section, you'll notice that the areas where carbon 14 is will develop on the film, and then you compare this film to the sections under the microscope, and you'll notice that these developed sections coincide with the phloem tissue. The second piece of evidence is using aphids. So aphids are tiny insects that pierce the phloem tissue with their sucking mouth parts. And microscopic examination shows that this is the only place they pierce. So on the left there you can see all those tiny aphids on a stem. If you provide a plant with labeled carbon in the carbon dioxide, this carbon is going to be incorporated into the carbohydrates. These will be translocated throughout the phloem, and the sugars in the aphid honeydew will contain the labeled carbon. So this shows that the products of photosynthesis are in the phloem. Just something interesting. Ants and other insects feed on the sugar, not on the aphid as it might first appear. Now, because aphids are parasites, some gardeners use predators that do kill the aphids to control the population. You'll remember that pesticides are chemicals, systemic pesticides are chemicals that move through the phloem and then they're eaten by the predators. The third piece of evidence is ring barking. So what happens is, as you can see on the left there, an area of the stem of a photosynthesizing plant is destroyed using steam or by peeling off the outer stem. This is called ring barking. And the phloem lies below this, so the phloem is also removed. After a while, the stem above the damaged area swells because the sugars moving down will not be able to pass and they'll accumulate. So eventually ring barking will kill a plant. And it's a good way to kill unwanted invasive species. Ring barking can also be used to show phloem contents, how they move both up and down the stem. So if a leaf is given radioactive carbon, so the sugars will be labeled, these will move into the phloem. The phloem is damaged just below the leaf, and the labeled sugars are then found in the phloem above the leaf, showing the contents are moving upwards. Okay, finally, evidence for and against the pressure flow theory. Okay, supporting the evidence, pressure within sieve tubes results in sap being released when these sieve tubes are cut. The concentration of sucrose is higher in the leaves, the source in the roots, which is the sink. Downward flow in the phloem occurs in daylight, but it ceases when the leaves are shaded or at night. Metabolic poisons and or oxygen inhibit translocation of sucrose in phloem. And companion cells possess many mitochondria and readily produce ATP. Evidence against or questioning the pressure flow theory include the function of the sieve plates is unclear because these would prevent mass flow. So they might help with preventing the tubes from bursting under pressure. Not all solutes move at the same speed. They would do so if the movement was by pressure flow. And then lastly, sucrose is delivered at more or less the same rate to all regions, rather than going more quickly to the ones that are with the lowest sucrose. Here's a lovely diagram to show you the process of translocation. So I'd take a moment to draw it and see if you can describe what's occurring at each stage. Then you would need to write down the evidence supporting the movement of solutes and phloem. And that concludes our lesson. The end.